So yes, very good evening to all the NEET aspirants 2026. This is your Prasanna Ma'am from NEET World. So we are done with one chapter, that is cell the unit of life. Now we are going to discuss the major concepts from the chapter cell cycle and cell division. Okay, from this cell cycle and cell division, what are the concepts that we are going to exercise? We have divided it into three major concepts. First we are going to discuss about cell cycle, and the next class or the next concept that will be purely on mitosis, where I will be solving few numericals also related to mitosis, and third will be meiosis. Fine, third is meiosis. So in this way we will be moving on beta. Okay. So first let us understand what is cell cycle. What is cell cycle in your perspective, you tell me. What is cell cycle? Cell cycle is nothing but the sequential events that are happening in the cell, from genome duplication, duplication of cellular constituents, duplication of different organelles, and thereby producing two daughter cells. Okay. What is it? First, it is sequential events involving genome duplication. Duplication or replication is one and the same, remember, including genome duplication, increase in cell size, replication or duplication of cellular constituents and cell organelles, and at the end division of or formation of two daughter cells. This complete sequential events is called as what? Cell cycle. So cell cycle will be operated in two major phases beta. Look into it properly. What are those two major phases? First phase is called as interphase, second phase is called as M phase or mitotic phase. First phase is interphase and second phase is M phase. Who is this? Ma'am, is this for 11th class students? This is for 11th students, for 12th students, and for repeaters also. Okay, whoever are working out for NEET 2026, for all of them this sessions will be helpful, because these are picked top 100 topics for your NEET 2026. Okay. So yes, let us get back to the concept. So what are the two major phases for cell cycle? First is interphase and the second phase is what ma? M phase. Interphase will be having three subphases. First is G1 phase, second will be S phase, and third is G2 phase. Okay. And M phase is also of three types. What are they is amitosis, mitosis and meiosis. I will be telling you, see. So cell cycle is operated in how many phases ma? Cell cycle is operated in two phases. Yes. What are they? Interphase and M phase. Interphase has three subphases. What are they? Very good. G1, S and G2 phase. M phase is also of two types. Okay, three types actually. What are they? Amitosis. Very good. Mitosis and meiosis. In mitosis and meiosis, two substages will be there. Okay, division happens in what? Two subdivisions. Okay, it happens in two substages. What are they? Karyokinesis. Yes, karyokinesis and cytokinesis. Fine. Karyokinesis and cytokinesis takes place both in the mitosis and meiosis. Remember, in mitosis there is only one karyokinesis, that is only one nuclear division. Cytokinesis is what? Cytoplasm division. So there is one karyokinesis and one cytokinesis. When it comes to meiosis, Nana, meiosis you have two karyokinesis and cytokinesis. karyokinesis 1, cytokinesis 1, followed by an interkinesis. The space between karyokinesis 1 and 2 is called as what? Interkinesis. And after interkinesis, you will be having karyokinesis 2 and cytokinesis 2. Have you got the framework of cell cycle? Now let us dig a bit. Okay, G1 phase, S phase, G2 phase. Remember the sequence properly. Okay, first let us look into G1 phase is also called as post M phase or pre-synthesis phase. Okay, G1 phase is also called as what, Nana? Post M phase and pre-synthetic phase, pre-S phase. Same ways, S phase is also called as what? Post G1 phase and pre-G2 phase, post, say Kunal, post
G1 phase,
for G2 phase,
post S phase
and pre-M phase. Sanjana, you tell me better, for M phase what we can say? Post G2 phase and pre-G1 phase. Okay, it is after G2 phase, right? So, post G2 phase and pre-G1 phase. Clear with this, right? Now, let us look into some more points of interphase. Interphase is also called as resting phase, preparatory phase, or biosynthetic phase. Remember all the synonyms. Okay, it is called as resting phase, preparatory phase, or biosynthetic phase. Why is it called as resting phase? Because actual cell division is not happening over there. So, that is why it is called as what? Resting phase. Why is it called as preparatory phase? Because complete preparation for cell division is happening in the interphase, be it increase in cell size or duplication of the cell organelles or duplication of the nucleus. Okay, everything is happening in the interphase. That is why it is called as preparatory phase. It is called as biosynthetic phase because cell organelles, if there is one copy of cell organelles, two copies of cell organelles will be synthesized, right? If there is one copy of nucleus, two copies of nucleus will be synthesized, right? As it is involved in the synthesis of cellular constituents and other nuclear materials, it is called as what? Biosynthetic phase. Fine. And one major point about interphase is, interphase will occupy 95% of the cell cycle time. Okay, how much time it is occupying? 95%. Same like you people, right? You will be investing, some people like 10 months, some will be 12 months, some will be 2 years of your investment of your time on NEET preparation. How much time you will be writing the exam? Just 3 hours 20 minutes. So, preparation is long than execution. Same way for interphase it is 95% of the time or tenure is occupied in the cell cycle. Clear till here, right? Now, let us discuss about the G1 phase, and as it is 95% of the cell cycle, let us see in different cells. For human cell, the maximum time or the complete time of cell cycle will be how much? 24 hours. 24 hours. One cell to complete its complete cell cycle, that is interphase and M phase, it will take how much time, Nana? 24 hours. Of this 24 hours, 23 hours will be for, yes, interphase, and just 1 hour is for what? 1 hour is for M phase. Same in the case of yeast cell also, where the complete cell cycle will be for 90 minutes in the yeast cell. Clear? These are some basic points that you have to know. Now, let us look into each and every phase. Okay? First, G1 phase. In G1 phase of the interphase, what is happening? G1 phase of the interphase. Before going to G1 phase, let me look into these comments. Ma'am, how many days you will be teaching? This goes for 100 live sessions, beta. In 100 live sessions, 100 concepts will be covered. Clear, ma? Kapil Dev. And next, yeah, 100 live sessions. Coming back to the concept in G1 phase. Kunal. In G1 phase, cell size will be increasing. Most of the cell organelle duplication will happen in the G1 phase itself. What? Most of the cell organelle duplication, most of cell organelle duplication and cell size will be increased in the G1 phase. Cell size increases, most of the cell organelles duplication and all the cellular constituents which are required for the S phase are being synthesized in G1 phase itself. Okay? G1 phase, in G1 phase cells will be metabolically active. Okay? In G1 phase cells are metabolically active. Now, after G1 phase, where do cells drop into? What is the second phase of the G1 phase? S phase. But before going to S phase, how about few cells like nerve cells or heart cells? Are they continuously, listen first, like heart cells, nerve cells, will they continue the cell cycle or will they have repetitive cell cycles? No, they divide very rare or very often. Okay, very rarely they divide. So, those cells which divide very rarely, from G1 phase they will escape to G0 phase. Okay? G0 phase is called as what? Quiescent phase. Quiescent. It is not crescent, it is quiescent phase or quiescent stage. Okay? That is G0 phase. So, from which phase to which phase the cells will be moving? If they are into inactive stage, G1 to G0. Okay, G1 to G0. What, like, name few cells like which go for rare cell division? Okay, which are those cells? Heart cells and nerve cells from G1 to G0 phase. Permanent tissues also from plant cells. Okay, so they will be moving from G0 phase to G1 phase to G0 phase. This is called as what, ma? Quiescent stage. In this stage also cells are metabolically active, remember that. Okay, in this stage also cells are metabolically active. Fine, ma? And from G0 phase, if they are back to the cell division, as Kunal said, permanent cells. Name few permanent cells which get back to the cell division again. What do you call those cells as? I will tell you. Cork cambium or vascular cambium. Okay, these are the cells which are getting back their meristematic activity or divisional activity. So, they will get back from what? G0 phase back to the G1 phase. Clear? Keep this thing in your mind. From G1 they are moving back to the G0, from G0 again to the G1 phase. Clear right now? Now, so in G1 phase what are the major activities you will remember? Most of the organelle duplication and cell size will be increasing. Cells are metabolically active. And those cells which rarely divide will move to the quiescent stage, that is G0. Again, if they are back to the division, from G0 to G1 only they will come. Clear, right? Next, this you see S phase or synthetic phase. In this S phase or synthetic phase, two major activities you have to remember. What are those two major activities? Listen carefully. If you suppose this as a cell and this as a nucleus. Okay, in nucleus let us suppose this as one chromosome. I already told you when we are dealing with cell, the unit of life, one centromere means it is one chromosome. This is how many chromosomes? One chromosome. This is how many chromosomes? One chromosome. Of course, chromosome structure will be clearly visible in the metaphase, but for your understanding and to deliver one point clearly, that in S phase only DNA duplication happens, but chromosome duplication will not happen. Okay, so here if you see the DNA content, okay, the DNA content which is like of this one chromatid, you can take this as the duplicated DNA. Okay, so here you can clearly observe, right, DNA duplication happens but chromosome number remains same. Okay, remember that point. DNA, yes, but there is a hypothetical question which is given in your MCQs, and there is once asked previous year question also, a hypothetical question to make you clear that DNA is only duplicating, there is no chromosomal duplication is what they want to convey. Okay? So, only DNA duplication and there is no chromosomal duplication. Chromosomal duplication. Clear na beta? And there is one more thing happening in the cytoplasm. Inside nucleus, DNA duplication is happening. Inside cytoplasm, only in animal cell, one non-membrane bound organelle is present. What is that? Both offline and online students. One student responded, but keep quiet. Rest other students also. What is the only non-membrane organelle sticked to animal cell? Of course, it is centriole, right? Okay. This centriole is present inside what? Tell me. Centrosome. Centriole is present inside the centrosome like this, which is perpendicular to each other. Fine. So, this centrosome will be duplicated inside the cytoplasm during which phase? S phase. So, here let us suppose if one centrosome is present in the G1 phase. In G1 phase, one centrosome and two centrioles are present. Let us suppose. In S phase, after duplication, one more centrosome with two centrioles will be formed. Okay? So, G1 phase at the end of S phase, if this is the number in G1 phase, what will be the number at the end of S phase? 2 centrosomes and 4 centrioles. Keep that in your mind. Okay? And there is one major protein synthesis happens in the S phase. That proteins are histone proteins. Histone proteins are synthesized. Histone proteins are synthesized during which phase? S phase. Why histone proteins are synthesized? As DNA is getting duplicated, listen first, as DNA is getting duplicated, to keep that DNA in the same space of the nucleus, it need to get condensed. For the condensation of DNA, histone proteins are required, right? That is why DNA duplication, centriole duplication and histone protein synthesis will be happening in which phase? S phase. Clear, right? Both of you, fine? And online students also. So, yes, in the nucleus during S phase, what is duplicated? Inside the nucleus during S phase, DNA is duplicated. Inside cytoplasm, what are duplicated? Centrioles. And centrioles or centrosomes, and what proteins are being synthesized? Histone proteins are synthesized. That's all with S phase. Coming to G2 phase, in G2 phase, major organelles like, you can remember with this mnemonic, MCG or CMG, any mnemonic you want, like what are the organelles that are duplicated? Mitochondria, very good, mitochondria, chloroplast and Golgi apparatus or Golgi bodies. Among these three organelles, which organelles do you include under semi-autonomous or self-duplicating organelles? Mitochondria and chloroplast. So, if there is a question to you, which is the phase, like what is the phase in which self-duplicating organelles are replicated? G2 phase. It is as simple as it is. So, these two organelles, mitochondria and chloroplast, will get duplicated in which phase? G2 phase. Okay, these are semi-autonomous, yes, semi-autonomous or self-duplicating organelles. Self-duplicating. Clear right now, beta, with all the three phases? And whichever more protein constituents or RNA, whichever are required additionally to conduct the M phase, are being produced in what? G2 phase. Okay? And so, now you are clear with the interphase, right? In interphase, you have G1. Tell me the synonyms of G1 phase once again. Post S phase synonyms? So, in G1 phase, protein molecules are synthesized, most of the organelle duplication is taking place, cell size is increasing. Whether everything has done properly or not is being checked by checkpoints. Okay. So, there are two checkpoints here beta, one is in between G1 and S phase, you call this as major checkpoint, and there is one more checkpoint between G2 phase and M phase. What is this checkpoint called as? Minor checkpoint. Okay. Major checkpoint will be having G1 cyclins and CDC2 kinase. These are protein molecules. Okay. G1 cyclin and CDC2 kinase will be having in what? Major checkpoint. They will check whether everything has properly done or not. Same ways, in S phase what is happening inside the nucleus? DNA duplication. Inside the cytoplasm what is happening? Centrosome duplication or centriole duplication and histone protein synthesis. Is that done properly or not? And in G2 phase, the self duplicating organelles and Golgi bodies are being duplicated. That is being checked by the minor checkpoint by what? You know, M cyclin and CDC2 kinase. Clear, right? So, is everything clear with respect to interphase? Fine, no? So, I have used to use something called as in cell division. In cell division, I have taught you, amitosis, mitosis and meiosis. Do you have any idea about amitosis? Amitosis, mitosis and meiosis. After this, like this, two daughter cells will be formed. Amitosis is nothing but binary fission beta. Binary fission. Tell me in few unicellular organisms where this occurs. Binary fission like amoeba, paramecium, euglena, where longitudinal binary fission occurs in euglena. Binary fission is called as amitosis because here in this division there is no involvement of spindle apparatus formation. Okay, there is no involvement of spindle apparatus formation. Just the nucleus, cytoplasm and the cell membrane will be dividing into two. Fine. Without the involvement of spindle apparatus formation, that is why it is called as what? Amitosis. And we have discussed like in mitosis, sequential events will happen. What are they? Karyokinesis followed by cytokinesis. Right now, beta? Karyokinesis followed by cytokinesis. Karyokinesis will be having 4 substages. Okay. What are they? Yes. Prophase, metaphase, anaphase and telophase. These are the 4 substages under what? Karyokinesis. Under cytokinesis, if it is a plant cell, okay, centripetal way of cell wall formation takes place or cell division takes place, cytoplasm division. If the animal cell is present, through the cell furrow it is centrifugal. Okay. So, just look into that, animal cell through cell furrow and plant cell wall is formed through what? Cell plate. I will explain you clearly this concept tomorrow. And in meiosis, as we have discussed, we have what? Karyokinesis 1 followed by cytokinesis 1. This is followed by what? Interkinesis and then again karyokinesis 2 and cytokinesis 2. In karyokinesis 1, you have 4 substages again. What are they? Prophase 1, metaphase 1, anaphase 1, telophase 1. The major important segment here is prophase 1, where it has 5 substages will be there. Definitely in every alternate year of NEET examination, you can see one question in the 5 substages of prophase 1. That is very important segment. I can tell you that is the heart of this chapter. Okay, wherein you can see from prophase 1, you have what are those 5 substages, Nana? Yes, first is leptotene, zygotine, pachytene, diplotene and diakinesis. Very good, ma. Okay, these are the 5 substages. In each substage, what is the event that is happening should be memorized by you. I will be telling all the mnemonics to remember, no issues at all. And here you see karyokinesis 2, again it has 4 substages, prophase 2, yes, metaphase 2, anaphase 2 and telophase 2, same as that of mitosis. Okay. And mitosis is called as equational division.
Ploid is maintained.
Yes, ploidy is maintained and it is called as equational division, and this is called as reductional division, where the chromosome number gets half in the daughter cell. Here the chromosome number remains same, that is why it is called as equational division. Equational division because chromosome number remains same. This is called as reductional division because chromosome number gets half. Clear? This is all about interphase and the introduction of mitosis and meiosis. Let us learn mitosis and meiosis in a unique way, where we will complete the concept, understand it well, apply the concept, and then we will be looking into some numerical applications as well. Clear, Navita? Yes, done with today's class and we will meet tomorrow.